Today we are going to discuss about a new topic, which is the bacterial appendages. First, we need to understand what are appendages. As we are having appendages of uh, animals, let's suppose insects, they have appendages which are legs. They have appendages wings. We have our appendages hands and foot and such stuff. Bacteria also has appendages, which is basically known as the flagella, fimbriae, and pili. There are three major appendages which are found in bacteria, among which number one is flagella. Flagella basically it is a slender whip-like extension, which is originated from the cytoplasm. In eukaryotic flagella, we were having uh, the flagellum being originated from the basal body kinetosome. But since we are not having any basal body present, any sort of double membrane bound organelle present within the cytoplasm, the basal body formed over here is not kinetosome, it is rather a different type of basal body. In that case, the structure of the basal body is shown over here in the diagram, and we are going to discuss that as well. Moreover, it also in our basal body is also known as a motor portion, from where the actual movement is generated, actual energy is generated in order to spin, in order to rotate the flagella. And after that there's a hook region. Hook is basically a curled region or curved region, which basically originates from the cell membrane. It does not originate from the cytoplasm, it originates from the cell membrane. And it originates from the cell membrane and protrudes through the cell wall in gram positive bacteria, but in gram negative bacteria it doesn't protrude through the cell wall. The reason will be told later. After that, when it comes to the filament, the third part, filament is the third part, which is also known as helical screw. And this helical screw basically is responsible for turning around, just like a screw. If we talk about a screw is having a shape like this. I mean, a screw is supposed to have a helical shape. Whenever we are going to move it with the help of sort of a screwdriver, then a screwdriver causes to rotate it at a 360-degree angle. So at that moment, the same movement will occur as if we were screwing our a screw through the screwdriver. One more thing over here, that here we are also having a hook present. And one more thing, that here when it comes to the vessel body basically is having vessel body basically it is consisting of several rings. Several rings are there in the vessel body. We are going to discuss that right now. Here we are having four major rings. uh, In gram positive bacteria, there is one pair of ring, and in gram negative bacteria, there are two pairs of rings. When it comes to the first part, we are not going to discuss the diagram here. We are just going to discuss the main components. Here, this red color ring. So let me just explain it a bit. Here, this is a cell membrane. This is what we call as a cell membrane. After which there is a peptidoglycan layer, and after that we are having the outer membrane. It's the structure of a gram negative bacteria, as previously told. Now here this purple color structure, red color structure, which is given over here, which is found on the cell membrane. This structure is known as a ring, which is known as SM ring. SM ring or MS ring, both can be called. Here it is basically a pair of two rings, which includes the S ring, which is a supramembranous ring, and second includes the M ring, which is a membranous ring. When it comes to the supramembranous ring, it is the ring which is found in the cell membrane, but it is a bit protruded outside the cell membrane in the periplasmic space. But when it comes to the membranous ring is anchored fully into the cell membrane. After which we are having peptidoglycan layer. In peptidoglycan layer, in case of gram negative bacteria, we are having two additional um, rings which are associated with the cell wall region, while in gram negative bacteria there are two rings additionally associated with cell wall region, but in gram positive bacteria there is SM ring only associated with cell membrane region, and there are no rings found associated with cell wall region. So in peptidoglycan cell wall, peptidoglycan, what do we call it? Peptidoglycan cell wall region, a cell wall layer, we are having a ring which is known as P ring. Now, what is meant by a P ring? Here you can see this is the brown color one P ring, and that basically is named on a peptidoglycan layer in which it is present, after which we are having the L ring which is found in the outer membrane. L represents lipopolysaccharide or lipoprotein which is present majorly within the outer membrane, after which we are having a rod. Now all of these rings are connected with each other by a central rod. All of these rings are connected with each other by a central rod. These discs or rings are connected with each other by a central rod, and after that we are having a structure uh, of hook structure which is known as a curled structure or curved structure, which is found larger in size. This curved structure is quite thickened in size, and this structure is what we this curved structure basically is what we call as hook, and this hook here is particularly this hook here particularly is responsible for the origination of the filament. Now, for the filament as well as for the attachment of the vessel body with the filament. One more thing, that within the vessel body motor portion, we are having another ring which is found in the cytoplasmic region, and that ring is known as C ring, which is also known as ratter of motor. Ratter of motor. We are going to discuss in our next topic of motility that how does it actually work, and uh, after that we are having the hook, and that hook basically it is longer in gram positive bacteria and shorter in gram negative bacteria. Reason behind it is that in gram positive bacteria we are having only one pair of rings, as a result of which the gram positive bacteria, let's suppose if we are supposed to draw the structure over here, gram positive bacteria, let's suppose this is a rough structure of cell membrane over here, after which we are having another layer of peptidoglycan. So in gram-positive bacteria, we are having only two rings, which are known as S and M rings, such that these S and M rings, when will originate the hook outside it, the hook will be originated from the cell surface. As a result of the hook being originated from the cell surface, will be originated from the cell membrane, because the rings are found only in the cell membrane and are associated with the cell membranic region rather than the cell wall region, due to which being associated with the cell wall uh, being associated with the cell membrane region and not with the cell wall region, we would be having the hook being originated from the cell membranic region, due to which being originated from the cell membranic region, this hook will be required to protrude through the cell membrane, will be required to protrude through the cell membrane, as a result of which it needs to be a bit longer in order to penetrate through the peptidoglycan layer of the gram positive bacteria. But when it comes to the gram negative bacteria, we are all having four rings which are also uh, associated with cell wall, such that two of the outer rings are associated with cell wall, which includes the L ring and P ring. So the hook here will be originated from the L ring or from the outer membrane or protruded from the cell wall rather than the cell membrane, due to which we'll be having uh, the hook being formed from the cell wall region rather than cell membranic region, or in other words, it is originated from the cell surface region, due to which being originated from the cell surface region, it is now basically being originated from the cell surface region, cell wall region, it needs to be a bit shorter because it does not need to protrude any other layer. So in this case, gram-negative bacteria, due to this reason, is smaller hook having, but a gram-negative, gram-positive bacteria is having a larger hook, right? And after that, when it comes to some other characteristics, over here we are having gram-positive and gram-negative bacteria. We are having certain things such that in gram-positive bacteria, again, the hook will be originated from the cell membrane rather than the gram-negative bacteria, in which the hook is originated from the cell wall. So after that, we are having the filament. Filament basically performs a rotatory motion. It, unlike the uh, you know unlike the eukaryotic flagellar motion, there is a 360-degree rotation of the filament, such that it rotates 360 degree, such that as it rotates 360 degree, due to the movement of the motors present, it causes the 360 degree rotation of the filament, as a result of which the 360-degree rotation of the filament ultimately leads to the movement of the entire bacterial cell, due to which the bacterial cell starts spinning and moving in a spinning motion, due to which in this manner the spinning movement will occur, such that when there would be a 360-degree rotation due to the motor's energy generated, the 360-degree rotation will cause the cell attached to the flagellum to rotate as well, as a result of which the cell will rotate ultimately, or in other words, spin, due to the rotation of the flagellum, due to which it will spin and move forward due to the movement of flagellum. Apart from which, when it comes to another structure over here, which is known as pili. Pili, basically it is a tubular extension. It is a tubular extension of the cell membrane. It is originated from the cell membrane, and it protrudes out through the cell wall, and it is basically formed of a globular protein which is known as pillin, whereas the filament of the flagella is formed of the globular protein flagellin rather than microtubules. No microtubules are present, rather flagellin are present. After that, when it comes to the pili basically involves pillin protein. As you can see in the diagram as well, there is pep A, which is having greater than 1000 pillin proteins, after which pep K, then pep E, then pep F, and then pep G. We're not going into detail of this. After which, when it comes to some other characteristics, as you can see in the diagram, it also showcases us that here the pili is originated from the cell membrane and is continuous through the cell wall. After that, it is shorter in size as compared to flagella, such that flagella are long appendages, pili are short appendages. Flagella are 20 nanometer in diameter and 20 micrometer in length, whereas pili are much shorter. While when it comes to flagella, these are hollow. Flagella basically are non flagella are helical and non-hollow structures. Flagella are helical and non-hollow structure, whereas pili are non-helical and hollow structures. So this is a point to be considered as well. After which, we are having another structure similar to pili, which is known as fimbriae. One more thing, that pili is found in gram-negative bacteria only, whereas the flagella is found in both gram-positive as well as gram-negative bacteria. Fimbriae is found in gram-positive as well as gram-negative bacteria. Fimbriae is also made up of the pillin protein, but here the fimbriae basically is you know are slender extensions. These are basically having structure which are similar to pili. The only difference come in its size, such that it is a bit smaller than pili, such that pili is associated with pillin protein, and the fimbriae is also associated with pillin protein. However, when it comes to the function, the pili protein is the pili protein When it comes to the function, the pili is particularly responsible for conjugation, which is a process in which the bacteria transfers genetic material from one cell to another. Whereas when it comes to the fimbriae, it is associated with the attachment of the bacterial cell on the tissue of an infected person or any other living inert surface. After which fimbriae is smaller in size and it is found larger in number. Villi are found fewer in number. And on the other hand side, when it comes to fimbriae basically is found smaller in size and here. Fimbriae is also a hollow and non-helical structure. All right. And after that, so these are some characteristics about fimbriae, pili, and flagella, which are the locomotory or appendages. And pili are also certainly found in a few species, particular species of gram-negative bacteria rather than the entire gram-negative bacteria. And apart from that, we are also having fimbriae and pili responsible for some other functions, such as pili is responsible for resistance of the bacterial cell from other microorganisms as well. So this is all about the video. Hopefully we meet again.